Welcome to Brain Vision.
Hello students, let us solve this question. In this question, it has been given that we have a mixture of amino acids and we have to analyze them. To analyze them, we have to separate them using ion exchange chromatography. Using ion exchange chromatography. Okay. And in ion exchange chromatography, specifically cation exchange chromatography, in which you have the sulfonate group. Sulfonate group, right? And it has been given to us that you have the buffer pH is 7, okay? And we have to tell which of from the mixture of amino acid which will be eluted first. So actually the one with the lower net charge or a negative net charge will be eluted first, okay? So now the first or A bit that is given to us is aspargine and lysine. We have to tell between aspargine and lysine which will be eluted first. So look at the structure of aspargine and lysine first. So if you look at the structure of aspargine, okay, so this is the structure of aspargine. Here you have an OH, you have an O here, you have an O here, you have an amine group here, and you have an OH here, right? And when it comes to the structure of lysine, what do you have? You have this O here, OH here, this is your carboxyl group, NH2, this is your amine group, NH2, right? Now, to see the net charge, we have to know the pKa values of these groups, of these functional groups. So here for this, the pKa value is 3.65. For this, the pKa value is what? 9.60. And for this, the pKa value is 1.88. In lysine, here the pKa value is what? 6. For this, the pKa value is 9.17. For OH, the pKa value is 2.18. So, if the pKa value, okay, if the pKa value is greater than the pH of the solution or the buffer, then protonation will happen. Protonation meaning hydrogen will be added. For example, the pH in this case is given to us as 7, right? And if you look at the pKa value of the NH2 here, so what will happen? The pKa value is 9.60, which is greater than the pH, that is 7. So it will get protonated, meaning one hydrogen will be added and this will become positively charged, right? Second case scenario, what can happen? Value of pKa is less than the pH. Then what is going to happen? It will lose the hydrogen H plus. So in case of this, when you look at the carboxyl group here, it is less than the pH. So what is going to happen? It is going to lose its hydrogen and this will become O minus. Similarly here, this will also become O minus and it has lost its hydrogen. So when you are calculating the net charge, when we are calculating the net charge, in this case, you have one negative, one positive, one negative. So net charge is what? -1. In case of lysine, if you see, what is going to happen? This will become plus 3 here and this will become O minus as hydrogen will be removed. So what will be the net charge here? The net charge is going to be +1 -1. That will give us 0. So you have 0 net charge. So from this information, we can tell that it is aspargine that will be eluted first. Why aspargine? Because it has a negative charge. So the amino acid which has a negative charge or which has lesser charge than the other one will be eluted first. So in part B, we have to tell between arginine and methionine, which is going to be eluted first. Now we'll draw the structure of arginine first. You have, this is the side chain of arginine, NH2 here, and you have another NH2 here. You have NH here, then you have your carboxyl, terminal carboxyl group, and you have an amine group. So we know that this is going to be positive, right? This will be our negative and this will be our positive because it, instead of being NH, it is NH2. Now, if you look at the structure of methionine, so you have, in methionine, you have a sulfur, right? Here you have your amine group, then you have your carboxyl group. So your amine group will be positive. Now, if we calculate the net charge, let us calculate the net charge. For this, the net charge is 1 and net charge in case of methionine is 0. So which will be eluted first? The one with lesser net charge, which is the one with lesser net charge, which is methionine. So your methionine will be eluted first. Now we'll move on to C. So in C, what do you have? You have glutamine, and valine. So again, we have to quickly draw the structure. So here you have the carboxyl group of glutamine. You have an amine group, another carboxyl group, right? So this is positive, negative, negative, right? And then structure of valine. Here is your amine group. This is your carboxyl group. So what will be the net charges? If you look at the net charge, in case of glutamine, the net charge is -1. In case of valine, the net charge is 0. So which will be eluted first? Of course, glutamine will be eluted first. D bit, which is glycine and leucine. So we have glycine and leucine. So if you look at the structure of glycine, you have your amine group here. Glycine has only hydrogen as the side chain, O, O minus. So here you get a positive charge. Here you get a negative charge. Now the structure of leucine. This is the structure of leucine. Here you have your amine group. So this is your glycine. Now let us write the net charge for both of them. Here you have one negative, one positive. So what is the net charge? 0. Here also you have one positive and one negative. So net charge becomes 0. Now when the net charge is 0, the heavier amino acid or the one with larger side chain is going to be eluted first. So you can see lysine has a large side group, whereas in glycine you only have hydrogen as a side chain. Therefore leucine will be eluted first. Thank you.